All right, so we're going to talk about the different energy pathways that we talk about in exercise physiology, right? We have our three ones, which is our phosphagen system, our ATP-PCr system, glycolysis, and then our TCA cycle with our oxidative phosphorylation in the mitochondria. Right now, let's just focus on our phosphagen system. So this one is going to include two main parts. So we'll just put our phosphagen, and this is our ATP and our PCR, right, our phosphocreatine. And this system is like immediately ready, right? We kind of break this out even to two if we want to, right? Our very first one is we have our ATP levels, right? So we have a concentration of, let's say between 7 to 8 millimolar. It kind of depends if it's dry muscle mass or whatnot. So this is our concentration. It's immediately there, and it doesn't last for very long, right? When you exercise like a max exercise or whatever, you'd only have enough ATP to last for like less than about 2 seconds or so. Like it's going to be, that's an S, for 2 seconds. It's like 8 muscle twitches and it's all gone. But remember what I talked about earlier with the steady state activation, it's not actually all gone because you quickly activate your other systems to maintain your ATP levels. And so I'm just saying that you only have this much ATP there. So if you didn't get the high flux going on, then your ATP levels would be gone. But in reality, under that max exercise, you're only using around 20 to 25% of your ATP, and that means that you have 75 to 80% remains at this max exercise. So you actually have quite a bit of ATP there. Obviously, then ATP is not the reason for fatigue. Other things are, which we'll talk about later. But this system is ready immediately. Okay, as soon as you move, like the twitch, right, moving my arm, like that was the resting ATP that's already there, and then as soon as that ATP decreases, that's going to activate the other enzymes, such as our creatine phosphate system, right? So you have your creatine phosphate. And what creatine phosphate is going to do is going to take creatine phosphate. Well, actually, I'll just write. There's a proton, but I want to write this first. Your ADP, which you just barely used, plus a hydrogen ion, and it's going to make your creatine. Now you just get creatine because you remove that phosphate, plus ATP. Okay, so this is the first thing that happens because you just know your ATP hydrolysis is going to get converted to ADP plus inorganic phosphate. Now, actually, this reaction with creatine phosphate is a reversible reaction. And this has a delta G prime naught of 12.5 kilojoules per mole. That's negative. So it is a favorable reaction, but it's not super high. And of course, it is higher when you have really higher levels of creatine phosphate and higher levels of ADP. And then this is going to make the reaction very favorable. But it is a very reversible reaction. But there's a couple of critical things that I want you to pay attention here is, number one, as soon as you break down your ATP, you get ADP, and then ADP gets immediately sucked up by the creatine phosphate reaction to using an ADP and a hydrogen ion. We know that hydrogen ions play a role in fatigue and a number of other areas. So this is kind of a very important and neat reaction that we're actually buffering some of those hydrogen ions, because the hydrogen ions are required to help to stabilize some of the ATP. So you get a buffering effect, and it depends on the pH as well. Of course, if the muscles are more acidic, then the pKa of the ATP levels are going to allow it to. It's going to alter its buffering capacity, right? So there's things about that too. But this is a very important reaction, and it's favorable reaction, and it happens very quickly. It's like this. We could say that this reaction with the creatine phosphate reaction is going to be ready immediately. That's an R, and it's going to last for we can say 10 to let's say 12 seconds or so. This is how long its duration. Okay, So that sprint, the 100-meter sprint that you're doing, this is the creatine phosphate system. And you can use quite a bit of your creatine phosphate. You can deplete it to around 70% or so. So you only have around, let's say 25, 30% remains. So this is like almost the opposite of how much you can deplete ATP, right? Creatine phosphate, you can deplete a lot of that because it's not as critical, of course, as maintaining the high ATP levels. So the enzyme that does this, we'll just put here CK, all right? This is a creatine kinase. And there's a number of different isozymes of creatine kinase, and that's why people have like a heart attack or muscle damage or whatever. You can actually look to say, okay, this is myocardial creatine kinase, and we can see if it's from the heart or if it's muscle damage or where it's coming from, because the creatine kinase enzymes are located inside the muscle fibers themselves. So if we look, like for example, you have creatine kinase can be one of them is going to be like on the M line of the sarcomere. And that's going to be important because that's where you have all the circuit pumps at, right? Now, the circuit pumps are, of course, are using a lot of your ATP very quickly because they are got to pump that calcium because it's binding onto troponin and it's allowing the contraction to happen. You want to have a fast relaxation time, and so you want to be able to quickly have remove that calcium from the troponin so that the myosin heads are able to release from the actin filaments. So that's your importance of having the creatine kinase right next to the circuit pumps on the M line. But you have another creatine kinase on there as well. Another important, I guess, concentration of creatine kinase, and this we can say is on the inner mitochondrial membrane. Okay, inner mitochondrial membrane. I don't know, space, I guess I can just say. And this one is important because it's going to couple your oxidative phosphorylation to your, basically to this one right here. And we think about what's happening. First on this one, you understand what's happening very, very easily because you're using ATP a lot right here. And so this reaction is going to happen. As soon as you use ATP, then you make ADP, and then the ADP gets replenished back to ATP, and then the ATP can be used here. Now over here, where you on the inner mitochondrial membrane, you get a whole bunch of ATP is being made in the ETC, right, in the electron transport chain, and this ATP is actually going to replenish this. We can go just like this. It's going to come here and replenish the creatine phosphate, driving the reaction this way. So you make a whole bunch of creatine phosphate, and then the creatine phosphate is able to shuttle that energy over to the ADP where your circuit pumps are, where you're actually using all that energy. If you think about it, ATP is a large molecule. Okay, so ATP is large. And it doesn't diffuse the diffusion. It has a low diffusion, right? Compare that to creatine phosphate, right? It's smaller and higher diffusion. And so by having the creatine kinase in the inner mitochondrial membrane space, you are making a whole bunch of ATP that's coming out of that ATP-ADP translocator. And the ATP comes out, immediately hits this enzyme, takes the ATP and the creatine, and makes ADP and creatine phosphate. The creatine phosphate then gets shuttled over to the circuit pumps or the ATP, the myosin ATPase enzymes, and then you have your creatine kinase, where your M line, and it's able to quickly resynthesize the ADP that's being used there and make your ATP, and then the creatine, it's just this whole cycle that's going back. If you think about it like this, you can say that ATP is mainly used locally. Okay, is mainly used locally versus versus your creatine phosphate. That's it can be shuttled, we can say. Okay. Because of course it can diffuse better. So I'm just trying to help you understand why this is such an elegant system by linking these two pathways together, where you can quickly replenish your ATP levels in this way. And the other benefit I wanted to mention too is, in order to drive mitochondrial respiration, you want to have ADP. Well, high levels of ADP, they actually activate the electron transport chain. Well, specifically they activate your ATP synthase enzyme. Okay, so that's really what's going on. And if you have high ATP synthase, then you get higher ATP levels. So think about what's happening. You get the ATP comes out of the translocator there at the membrane. Immediately, your creatine kinase here on the inner membrane space takes that ATP and converts it to ADP and creatine phosphate. The creatine phosphate goes over to the myosin ATPases and circuit pumps and to where the M-line protein is at, right? That creatine kinase converts that creatine phosphate to ADP and ATP, and then it gets used there. The ADP that gets converted by this reaction going this way, so there'd be this ADP over here, it happens right there on that translocator. So the ADP doesn't have to diffuse very far at all, right? It comes out, the ATP comes out, gets converted to ATP. The ATP comes out, gets converted to ADP, and then the ADP goes back in. So then it can go to the ATP synthase enzyme, gets converted back to ATP, and this keeps things it happens much faster, right? This is the kind of the neat thing about this. Now, this is also why creatine supplementation can be beneficial, because like at rest, let's say most people on average, right, you have how much creatine levels do you have, right? So a concentration, and it's gonna vary quite a bit. But we can say between say 80 to 120 millimolar of creatine. That's just naturally in your body. And if you supplement with creatine, okay, so supplementation, then it can increase this, supplementation. So we can just go supplement. Okay, it can increase it to say 140 to 180 millimolar. Okay, so now we have a much higher creatine concentration. Now the creatine, when it's stored in the body, you have about 2/3 of it is stored as creatine phosphate, and so 1/3 is going to be stored as just your creatine. So first, the creatine comes into your cells. It first gets phosphorylated, and it's stored mainly as the phosphorylated kind, which is exactly what you want, because you want to drive this reaction, right? You want to be able to maintain high ATP levels, and then the rest is stored as just creatine. So supplementation, typically, you'd want to take. Your body already makes around 2 grams of creatine a day, and then by dietary sources, you might get another gram or 2, depending on how much meat you're eating. But supplementation, you want to take around 5 to 10 grams of creatine a day. So 5 to 10 grams per day. And it actually depends, because if you're just looking to saturate the muscles, then even taking a slightly less than 5, looks like an S, right? But taking even less than 5 grams is even okay. But the reason why I wrote 10 and potentially even higher is because there's a lot of benefits for creatine on the brain and the whole central nervous system as well, because of this entire pathway. First, the brain already is using a lot of glucose because it doesn't metabolize fatty acids very well, and you have to maintain high ATP levels in the brain. That's so important, and well, for the whole body, but of course the brain is very important. And if you don't do that, then you can have all sorts of problems. Now, of course, the brain is very good at doing oxidative phosphorylation with pyruvate substrates from the end product of glycolysis, even consuming lactate. The brain loves to consume lactate as well. And that's all done by the oxidative phosphorylation process. And so by coupling this together, you're able to help to maintain your high ATP levels for your brain. But because you have to get through the blood-brain barrier, a number of things, you have to take higher levels of the creatine in order to start seeing increases in the brain itself. So there's quite a bit of research on that. And again, it's just amino acid. There's a lot of safety data on taking creatine. Sometimes I'll just add though that sometimes people who take extra creatine, then it gets broken down, it gets cyclized. So we could draw the shape of creatine, but it ends up getting it goes through a cyclization. And there's an electrophilic portion, does a nucleophilic attack over here, and that forms a creatinine. And then creatinine is a marker that's often used for kidney problems. But it's not creatinine itself is not necessarily harmful or causing damage. It's just that it's a marker of your kidney function, because your kidneys are supposed to operate at a specific glomerular filtration rate, and creatine breakdown is very constant in the body under normal conditions. But if you are taking extra creatine, then for some people it can slightly elevate the creatinine, and then it can raise alarms like, oh, something wrong with your kidney. But actually, that's not necessarily what it means. You have to use a different marker like inulin or something. But anyway, that's a different topic. But again, just focus on the importance of creatine itself. It is very important, and for this pathway, it's very important. And it can last, 10, 12, maybe up to 15 seconds, depending on your stores. You are going to have higher levels in your type 2 muscle fibers, all right? Because, well, why do you think that is, right? Of course, it's because you have, these are more glycolytic, right? So you have a higher levels of creatine kinase is going to be in your type 2 muscle fibers. And versus your type 1, of course, it has that in there as well, but thus more of your oxidative phosphorylation. Now, one thing that's interesting too, and you can try this yourself. You can really see how this works. Do like the wind gate you've seen in class before, or you can just try this on your own. Try to sprint as fast as you can for as long as you can, and you'll see for that first 5 to 10 seconds, you feel like you could run forever. It's like, yeah, I got this. This is no problem. But then quickly, just a few seconds after that, you will feel an immense amount of fatigue and everything. And that sort of shows where your creatine phosphate was primarily being used at. I mean, your creatine phosphate, with that system, you literally can make 8 to 10 millimoles per kilogram of ATP per second. And you compare that to, say, glycolysis, that's only 4 to 5 millimole per kilogram per second, which is still a lot. It's double as high as the oxidative phosphorylation. But the amount of ATP that can generate is very high. So it's just an interesting correlation that as you sprint and run that hard, you can literally tell that you don't have much energy left. But again, it's not necessarily the depletion of either creatine phosphate or the ATP that is causing the fatigue. That's something else that's going on that just correlates with all of this together. Now, another thing to maintain your ATP levels, though, is this is not really part of the creatine phosphate system. I'm just gonna erase this so we have a little more room. It's not really part of a creatine phosphate system, but it's another reaction that can happen. Is you can take your ADP that you're being produced from hydrolysis, right? And you can take 2 of these right here, and you can convert this to ATP and AMP. Now, AMP is a pretty cool molecule, right? Because what does this one do? What can AMP do? Well, AMP, it can activate what we talked about, AMP kinase, right? This is going to be the stimulus to activate your PGC1 alpha. This is a very potent stimulus, and one of the benefits of having high AMP levels. Another thing is it can activate your glycolytic enzyme, PFK1. There's another one, PFK2. This is it's not a glycolytic enzyme, but it's another enzyme that makes glucose phosphofructokinase 2 is an enzyme that makes another molecule, fructose 2,6-bisphosphate, that really activates PFK1 as well. But this is allosterically regulated. I can just write, maybe you can read it, allosteric. And it's going to increase PFK1 and PFK2, and that's going to give you the increase in glycolysis. Because you think, why would you want to increase glycolysis? Well, it's because you need to make a lot of your ATP. And then if we think about more what's going to happen, it can also activate your glycogen phosphorylase. Glycogen phosphorylase. And that, of course, also going to increase your ATP. It can also increase your GLUT4 translocation. And of course, so if you have, this is going to increase glycolysis too. So GLUT4, of course, is that transporter, as we talked about, that will take the glucose out of the blood and bring it into the cell, right? So it's a PI3 kinase mechanism as well. Now, so AMP is really doing a lot of really cool things. But another thing, though, that can happen if we're not careful is this AMP levels. As it gets higher, it can increase the levels of adenosine. And this can be problematic because adenosine can bind to these A1 receptors and on the like on the muscles and the neurons, and that's going to lead to fatigue. Okay, neurons. That's going to lead to fatigue. Adenosine is this byproduct of your ATP metabolism. And in fact, there's other types of these receptors. There's one called the A2A receptor. And I'll just add that caffeine is an antagonist to this. And so that's why a lot of people, well, everybody, I guess, but this something that, so adenosine will actually bind to A2A receptors in the central nervous system and make you feel tired, make you feel sleepy. But caffeine acts as an antagonist to the A2A receptors, preventing adenosine to bind, and that's why caffeinated beverages can make you not feel asleep anymore because it has this mechanism. But that's on the A2A receptors. But adenosine targets on the A1 receptors, leading to some of that fatigue. So that's another issue, though, with building up AMP levels. We can get adenosine, and so there's other ways that we can clear the AMP as well. For example, you can take AMP and you can convert it to IMP. And if you when you convert to IMP, you also make ammonia. Now, this enzyme, this reaction here by AMP deaminase or a myoadenylate deaminase. Okay, I'll just put deaminase here. By this deaminase reaction. This reaction just basically takes off the amine group of the AMP, making IMP, but notice it forms ammonia. Well, this is very toxic to the cells, and this has a pKa of like 9.3 or so. So what's going to happen? It's going to immediately get protonated to ammonia, right? So this NH3 is going to pick up a proton from the solution, and it's going to form NH4+. We always circle our charges in organic chemistry. You can't even read that anymore. But it's Na4 ammonium. There we go. Circle that too. Okay. So now we have this is of course, ammonia is very toxic, but it quickly gets converted to ammonium. And so it's one neat thing about this, though, is because you are picking up a proton, you're helping to buffer all the hydrogen ions that are being created from ATP hydrolysis. Right? There is because there are hydrogen ions that stabilize this right here, and so when you do this, then you end up getting a hydrogen ion that gets produced as well from here, and so that's why ATP hydrolysis ends up creating protons, and these protons can be picked up by the breakdown of AMP to IMP. and then you form ammonia, and then and ammonium. And then ammonium can be recycled by in the urea cycle with different enzymes, and it basically gets excreted out. So that's one pathway. Also with AMP. Actually, we can continue going though, because once you do form AMP, for example, then you could actually recycle this back, and you can form. Well, you'll see it goes more into your energy pathways. So you can take your IMP and you can make your adenylosuccinate. I'm worried you're not able to read this anymore. But you're making adenylosuccinate here by adenylosuccinate synthetase, and this uses GTP, so there is some energy source that's being used here. But then you're able to get AMP. So now you're back to AMP. So AMP can do the same things that we talked about here, but you also form in the same reaction, you form fumarate. And fumarate can get converted to malate, and then that's going to enter into the TCA cycle. And so it becomes an energy substrate as well. So that's another potential pathway for AMP. And then I guess there's one other though, because you can't take IMP and when you break it down, you could also form inosine, and then you could form uric acid. And uric acid, you end up just, it goes out your urine. And it's metabolized by the liver, and of course high levels is toxic. But uric acid actually has an antioxidant activities as well. So it actually has some benefits, because when you're doing all these muscle contractions and mitochondrial respiration, you do produce reactive oxygen species, free radicals, and that can be picked up by some of the breakdown of inosine from IMP, uric acid, and then that can help to neutralize some of those toxic free radicals that would otherwise wreak havoc on the cells. So it's pretty elegant system that the body has to control all your metabolism. Okay. Now, of course, the big thing though that I want you to consider is you do decrease your ATP levels a bit. You are activating high levels of your AMP, and that way you're able to activate your PFK1, PFK2, and your glycogen phosphorylase, and you get glycolysis to really ramp up. And so now, and glycolysis, it actually starts very quickly. I mean, within just a couple of seconds, it starts, and it's reaching its peak very, very fast. But because glycolysis does not produce ATP at the same rate as the creatine phosphate system, well, that's why we have to really rely on this. But this pathway is going to help to activate your glycolysis system, which we're going to talk about next. So, all right, we'll go to glycolysis now.